Okay, well, welcome back, everybody. I hope you enjoyed this morning's session. My name is Scott Blanchard, and I am delighted to moderate this next session. Our first speaker in this next session is Dr. Kristen Kutmu from University of Michigan, who will be discussing her work on integrating LC-MS, MS, and nanopore approaches to directly sequence RNA. We're very excited that you made time for this. Kristen, thank you so much. Take the floor. It's yours.
Thank you so much, Scott, for the lovely introduction. Welcome everybody back to the afternoon session. I'm very excited to be here. And I have to admit, I am a little bit intimidated because if I am going to be honest, I am a biochemist and not a mass spectrometrist, and have just kind of come into this field the last 5 or 6 years ago, as my lab is studying how modifications in RNAs impact the translation process. And it became rapidly apparent to me as we launched these projects that we needed to have some more quantitative tools for directly measuring modification levels in RNAs. And coming from a chemistry background and being in a chemistry department, I said, oh, mass spectrometry must be the way to do this. And luckily, I was smart enough to know mass spec should work and ignorant enough to know how hard it would be to deal with these long negatively charged oligonucleotides. And so I'm going to share with you what has arisen fortuitously out of that ignorance in my ability to acquire some stellar students and collaborators that have been able to help my lab push these ideas forward. And I want to share with you where we're at this moment in time. So we've known about the existence of modifications in RNA since the dawn of RNA research in the 1950s, when some of the most prevalent modifications found across biology were first discovered. And we've heard a lot about these modifications and their significance throughout the day today. And these modifications have the ability to really impact every step in the life cycle of an RNA after it's been made, changing their structure and dynamics, splicing and maturation, stability, how they interact with other RNAs or other proteins to ultimately influence their function, particularly in systems like protein translation, where every single one of the RNAs involved can include a modification. So despite knowing that these modifications are doing something important, I like to think of it a lot as the sorts of functions that can be done in post-translational modifications, which the world has known for a very long time do very important things. However, despite knowing that these exist in RNAs, knowing that the enzymes that are linked to these modifications, when they're perturbed, cause disease, and knowing that there are, tens of thousands of modified sites in a single cell, we still don't really understand what individual sites of modification do. And that's really what my lab is interested in trying to define the molecular level consequences of individual modifications. And just to give you an idea, even though we've been studying tRNA and rRNA modifications since we more or less discovered the ribosome, we know what far less than 50% of those modified sites do. And this was alluded to earlier this morning when someone, I can't remember who, asked a question about the tRNA body modifications, for example. We still don't know exactly how they're contributing to protein synthesis. Additionally, the discovery that mRNAs can contain thousands of different modified sites. We know that in general, having an M6A around, for example, seems to impact the stability of an RNA, but knowing what the biological function of individual sites, much less even just the stoichiometry of the modifications in those positions, remains a big challenge for the field. And I think really emblematic of this was something that shocked me when I, as a long-term RNA biologist, went into Modomics and just counted the number of tRNA molecules and rRNA molecules in different organisms that we knew exactly where their sites of modification were, not just what the overall sort of smattering of modifications that are possible in those organisms, but precisely where they are, and they've been mapped. You can see that even for key model organisms, anything beyond really E. coli and S. cerevisiae, we don't have good tRNA or RNA maps for, making it really hard to figure out what the function of these individual sites are. And in humans, it's vastly more complicated. So, as I mentioned at the beginning, and as is the theme of this entire workshop, mass spectrometry seems like a great way for us to start to get that information, to start to be able to quantitatively figure out what is where and when it's there and how much exists. And so, in the first half of today, we heard a lot in particular about nucleoside analyses, which have been conducted very widely since the 90s. And these sorts of analyses are absolutely fantastic for discovering and identifying new RNA modifications and for quantifying the overall levels of modifications within a sample. Additionally, there's been a smaller group of people who have been working on directly sequencing RNAs by mass spectrometry. And this has been moving a little bit slower, just simply because it's much harder with the long oligonucleotides to get them to fly. It's also much more difficult because there are not good enzymes like proteases. The enzymes we have for chopping RNA into small bits that can fly robustly on a mass spectrometer are super nonspecific relative to things that you might think about in a proteomic space, something like trypsin or chymotrypsin that's going to give you the same peptide product all the time. As an RNA biologist, if you imagine putting RNase A into your sample, you should not expect a uniform set of things to come out that will give you mappable sizes of RNA. So far, the field, which has been going on for quite some time, has been primarily, though not solely, but primarily focused on sequencing either purified short RNAs. So for example, the sort of really powerful techniques we heard about from Tom Suzuki this morning, where he's purifying out individual tRNAs in these Herculean efforts and sequencing them one at a time, or semi-complex mixtures, which would be like one, for example, Moderna did this with one mRNA, where it took a single mRNA, it knew what the sequence was, and it was able to digest it down and look at it on the mass spectrometer. However, looking at more complex sequences, the kinds that a biologist like me might be interested in, has been really much more difficult. So when I started my lab, we were really doing ribosome studies with modified mRNAs and tRNAs. And I was interested in trying to understand these things quantitatively. And so I thought, nucleoside analysis will be good for getting us what is in our samples. But I really need to know where things are in order to be able to study them really robustly. And so we launched 2 programs in the lab together with an analytical chemist here in my department, Dr. Bob Kennedy, to both develop a robust nucleoside platform, which I am not going to talk about today, and to pursue the idea of RNA sequencing. So I very naively thought that RNA sequencing would be just like proteomics, right? We're going to chop things up, stick them on the mass spectrometer, and it will be no problem. I learned very quickly, along with my student, that is not the case. And so we decided at the beginning to start small, to use the approach where we're starting by looking at one purified short oligonucleotide at a time. We started off by purifying individual tRNAs, just as Professor Suzuki's lab does. And then we moved on with my first analytical student, Josh Jones, who really spearheaded nearly all of the work you're going to see today, my collaborator, Bob Kennedy, and a new student that's come in now to take over these projects from Josh and is making significant headway with E. coli RNA sequencing, Kaylee Simcox. The first thing we did with Josh was to look not only at tRNAs, but at individual siRNAs, because there's a lot of interest from industry. In this case, it was in conjunction with Eli Lilly, to be able to sequence these small therapeutic RNAs, all of which are highly modified. However, this is not as simple as sticking it on the mass spectrometer, because we must be able to chop them down into pieces that will fly. And chopping down highly modified oligonucleotides is not necessarily an easy task. It's not something that all nucleases are up to the task to do. So Josh optimized some digestion platforms for siRNAs that contained 2' fluoro and 2' methoxy groups, which are commonly found in these siRNA therapeutics. He used an ion pairing reagent and subjected his fragments to MSMS in an Orbitrap Lumos instrument and was able to sequence siRNAs. As you can see, he tried a variety of different partial degradation schemes and eventually hit on nuclease P1 being the most robust way to chop up these highly modified molecules. He then went on and was able to reproducibly sequence 3 siRNAs labeled here LR1, 8, and 15, all of which have different numbers of 2' methoxy modifications. And that was a great start, but for me as a biologist, it really wasn't getting us where we wanted to go. It was like, okay, so we can sequence one thing that we know what it is, that is outside of a biological context. So the next thing we wanted to do is we really wanted to try to look at total pools of tRNAs. We thought that this was naturally a relatively simple complex mixture for looking at RNA modifications. It was defined, particularly in S. cerevisiae and E. coli, where we should know for some of the technology development what it is we expected to get out. However, people have been trying to sequence tRNAs for a long time and have been having some limited success with it. But the hard thing about sequencing these directly is that long RNAs don't fly well on the mass spec, so you can't just shoot the whole thing on and therefore can't be analyzed. And again, there weren't good RNases for giving us predictable pieces that we could reproducibly sequence. And Josh tried this for a really long time. He spent a lot of time and effort chopping up tRNAs with limited digestions, which is what's generally done in the field, in order to get a ladder of different oligonucleotides that he could then sequence. When he did this, he could reproducibly get about 10 to 30% coverage, which is totally on par with what other people had seen when they were doing total tRNA sequencing. And then one day towards the end of his PhD, Josh walks into my office and he says, "Kristen, I'm an idiot." And I was like, "No, man, I don't think you're an idiot. What's going on?" He said, "Well, I'm thinking about this like an analytical chemist and not an RNA biologist. I'm just taking my samples and unfolding them and subjecting them to these digests. What if instead I use the natural structure in the tRNA and just folded my tRNAs prior to digestion, and then subjected them, put in RNases that only cleave single-stranded regions?" This was a very simple innovation, but it really changed the distribution of oligonucleotides that we got and gave us much more long, mappable pieces and allowed us to directly sequence total pools of S. cerevisiae tRNAs with pretty good coverage, where on average, overall, we had about 70% coverage of the tRNA molecules, and some molecules had up to 90% coverage. An example of how just this folding procedure prior to digestion increased our ability to map tRNAs is shown here, where you can see for this particular asparagine tRNA, if you completely digested it, as had been done most commonly before, you had a handful of oligos that you could detect. However, when Josh simply folded his tRNA before digesting it, he got much longer pieces that he was now able to confidently map back. This has given us 60 to 90% coverage on most tRNAs, and that's fantastic. Since then, Kaylee has started in lab, and she's gone and she's worked on now expanding this and extending it to mapping E. coli tRNAs. And in order to do this, she's incorporated a new commercially available enzyme, RNase 4. As she's done this, she was able to, again, get a different distribution of oligonucleotides, as you can see here by the LC, for which she could then sequence directly on the Orbitrap again. And combining all of these different digestions, she was able to greatly improve E. coli tRNA sequencing as well, where now we're getting between 70 and 100% coverage on most tRNAs in a single experiment. Another important thing to mention about this particular experiment is that Kaylee was also able to extend her observations to E. coli tRNAs we're purifying in-house because it turns out that they act a little bit differently than when you buy a tube of total tRNA from Sigma. And there's been some finagling in order to get all of our in-house preps to work well. However, that's going to allow us to ask a lot more biological questions now that we can use in-house purified tRNAs. And as you can see from these blue bars, we get pretty good coverage across the board. All right, so now we're at a place where I'm going to talk about how we've extended our LC-MS/MS to also incorporate nanopore sequencing in order to do some de novo sequencing, where we're not just looking for modifications that we already know exist, but we're trying to robustly identify new modifications in new molecules that haven't been mapped, which really I think is the biggest challenge for RNA mass spec. It's one thing to be able to say, yes, something I expect to be there is there, and it's another thing to do things de novo. In order to do this, we're incorporating a wide variety of information. The first information we're taking is we're taking nucleoside analysis, where we're seeing what's in our sample to begin with. We're then conducting nanopore sequencing experiments, which allow us not to see what's in a sample, but to see where a modification might be in a sample. We're then combining the information from our nucleoside analysis, where we know what's possible and we know where modifications can be, to help inform our analyses of our mass spectrometry data in order to rigorously and reproducibly detect specific modifications. We've done this and published on this already once together with David Garcia, Matane Jain, and Mark Aikitson, where we've looked at modifications in S. cerevisiae tRNAs and actually discovered some modifications on tRNAs that weren't previously predicted. However, we saw them by nanopore and were then able to directly verify them by LC-MS/MS. And even more, I think, interestingly, in the de novo space is that we sequenced the MS2 bacteriophage, which had not been reported to have modifications. So in this case, we began off by doing nucleoside analysis on the bacteriophage together with Blake Wiedenheft at Montana State. And when we did this, we were able to correctly, in the correct ratio, measure the canonical bases, A, C, G, U, in a quantitative way. And then we scanned for 51 different possible modifications in a nucleoside analysis. And we saw that on average, the amount of pseudo-U in our samples corresponded to 1 pseudo-U per MS2 molecule. We then went and directly sequenced our MS2 molecules by partially digesting MS2 with RNase T1, separating out the fragments by helic chromatography, and then sequencing them in a process that required no ion pairing reagents. And I just want to emphasize that I think that using no ion pairing reagents is a really important thing for the field to move towards doing, because it makes doing mass spectrometry accessible to the average biochemist like myself on a core instrument, as opposed to people who just have specialized instrumentation. So when Josh did this, he was able to sequence the MS2 bacteriophage with about 60% coverage, and really he did not see any modifications, which is not surprising because using sequencing, we can't see the isobaric pseudouridine modification. We then did nanopore sequencing, and we saw that when we ran de novo nanopore and looked at it by Tombow algorithms, that many, like 1,000-fold more modifications were called by nanopore algorithms. And we said, okay, if that's the case, that's not good, but let's maybe look at this more specialized modification algorithms that will only give us information on the modification we know from nucleoside analysis is there. So we went and we applied 2 algorithms that had been written specifically for looking at pseudouridine, NanoSu and NanoIP. And we looked at a number of sites that had been called as pseudouridine in these nanopore algorithms. And we then subjected them to an orthogonal biochemical technique CLAP to directly look and measure on a site-by-site basis the stoichiometry at each one of those proposed sites. And what we saw here is that about, 5/8 of the sites were actually real, and they all had low stoichiometries, all of which added up to about one pseudo-U per molecule. Okay, and I'm going to end my talk by just saying that I think we should continue to integrate all three of these different pipelines in order to really rigorously detect individual sites of modification if we're going as we move forward to trying to do de novo sequencing. And thank you for your time.
Wonderful. Thank you, Kristen. I think we have some questions from the audience that I'd like to present. The first question from an anonymous participant is the folding method producing robust results? Every time are you getting the same fragments? And maybe just describe the folding procedure and why it's introduced in the same time.
Yeah, so the folding in our hands, yes, it does give robust results. We do get reproducible results. And it's just like any other tRNA folding that one as a biochemist does on a regular basis. You just heat the nature and fold.
Does that mean you have denatured it before you refold, meaning like phage electrophoresis or something?
So we haven't. The procedure that we use is that we first run an FPLC in order to get tRNA fractions. We then do a prep HPLC in order to further purify those fractions so that we're just tRNA, and then we follow up from there.
Okay, we got a lot of questions appearing, so I gotta keep going. Do you think that other fragmentation techniques, such as EPD, would give you better identification by decongesting the MS-MS spectra?
Possibly, okay. But our issue has been less with the MS-MS spectra and more with getting things that are long enough to fly well enough that you get enough depth to be able to say anything. Great.
While tRNA digest with RNases produce longer fragments, how do you confirm the location? That's another question.
Right. So you can confirm the location because these have sequence context. Right. So you have to have we're sequencing. We're not doing nucleoside analysis. Things aren't being chopped up to nucleosides. These have sequence context. So
wonderful.
You directly sequence them.
Great. And finally, do you find the same sensitivity when you use helic separation compared to IP reverse phase separation for your oligonucleotide analysis?
No, it's definitely better with IP, but the disadvantage of IP is that I run everything on a core instrument, right? We have a triple quad in lab, but I don't have an Orbitrap. And so they won't let us run ion pairing reagents. I think no core I know of will let you run ion pairing reagents. And so helic enables us to have pretty good sensitivity and do it on any instrument. And I really just want to emphasize, I think that this is something that we as a community really need to think about, is if we want people to be able to do these sorts of measurements, we need to make it possible for people to use these techniques where they're at and not just have to own an Orbitrap in their own lab in order to be able to do it. And as I've started off in this field and sort of made my journey in, it's become really evident to me that there is fantastic analytical chemistry out there and there's fantastic biochemistry, but the two halves don't really talk to one another about what needs are. And I think one need for making these techniques more widely applicable, as opposed to something like nanopore, which a biochemist can understand, is to make it so that you can run them on a core instrument. And that means no IP reagents.
Wonderful. We probably should move on. I think there are questions in the chat that you might be able to get to that are hopefully easy to address. But in the interest of staying on time, I think we should move on to our next speaker. Kristen, thank you so much. Wonderful talk. Our next speaker is Dr. Shenglong Zhang, a member of our center, Grant, and he is at the State University of New York in Albany, and he's going to be presenting his message on next generation mass spec sequencing, unlocking RNA's true sequence and the first human RNome.
Hi, everyone. It's really a great pleasure to see all those experts here. I really had a good time learning those new technology. Today, I'm going to talk about the next generation mass spectrometry-based sequencing, a new platform we have been developing for exhaustive and unbiased sequencing of RNA and modifications. We hope this new platform can be used for unlocking RNA's true sequence and the first human RNA. For this audience, I don't need to say much about the Human Genome Project. It took 13 years, billions of dollars to generate the first sequence of our human genome. Now, a comparable project at a similar scale would be the Human RNA Project to draft the first sequence of all human RNA and their modifications. We can call it the Human Epitranscriptome Project or Second Human Genome Project for RNA. However, this project has not yet been explored much, mainly because we don't have a sequencing technology compared to some sequencing that was used for first Human Genome Project. In fact, we actually don't know the true sequence of RNA or RNA known. That's including sequence of not just full nucleotide base ACGU, but essentially all the possible modification, as many as more than 170 different types. It's not trivial, as you can see, to determine the true sequence even for one RNA. Imagine how challenging it could be to determine the true sequence of all human RNA. If we said human RNA known as a goal, there are probably two main strategies that can be used to sequence the first human RNA known. The first one is what we call the one modification approach. Focus on one modification, like M6A, and map it across all different RNA types, like messenger RNA, tRNA, ribosomal RNA, etc. That will lead to M6A epitranscriptome, hoping this can be used to extend it to a lot of modifications as well. The lot approach is what we call the all modification approaches. Sequencing all possible RNA modifications simultaneously, but only on one RNA, like tRNA. That would need to tRNA known, and with the hope that this method can be extended to a lot of RNA types. So which method is more feasible? We know the first, the one modification approach definitely is the most popular one. But if we look close, the method used to map our M6A is M6A specific. They can be used for sequencing a lot of modifications. You may think in using different methods with each method for one RNA modification type. Collectively, now there are about 12 different RNA modifications can be mapped either by Illumina-based RNA-seq or NALP-based RNA sequencing. But this is still a very small portion. Those individual method would have difficulty to really to cover all the modification in the human RNA. There's another approach has been overlooked, but seems more feasible. That's what we call the all modification approach. Sequence all possible RNA modifications simultaneously, even it's only in tRNA. That would need to tRNA known, and this would contribute about 10 or 20% of human RNA known. If we can further extend this method to sequence a lot of RNA, like ribosomal RNA or messenger RNA, and then we have an approach that we can be used to sequence first human RNA known. Of course, this method has to be tested vigorously. A key question here is how can we sequence all different types of modification together with this for nucleotides ACGU. This is why we propose mass spectrometry-based sequencing. We know this is more commonly accepted, but we feel strongly this is probably one of the most promising methods that could allow us to sequence all the RNA modifications. Because for sequencing, we need to have a signature tag. In terms of tag, nothing's better than mass. Mass is a natural tag, both for canonical and modified nucleotides. It would be difficult now for another method to determine all those RNA modification identity. You are wondering how can mass be used to sequence RNA? In fact, conventional mass now even have difficulty to sequence RNA de novo. From previous talk, you probably know RNA has been used to analyze RNA for decades, coupled with the LC, and they were considered as a golden standard for RNA modification analysis. However, to use those methods, typically they have to digest RNA to a single nucleoside, and they can't provide the location information. Or they need another methods to provide the sequence information. So that's why we're developing what we call the next generation mass spectrometry-based sequencing. We would like to empower mass so that they can be used for de novo sequencing of RNA. We want to develop this for high throughput. We also want to use this for quantitative mapping those partially RNA modifications. So how can we develop mass sequence of RNA? If we look at Sanger sequencing has de novo sequencing capacity because the Sanger sequencing is relying on a letter generated by Sanger dideoxy reaction. Of course, this is a Nobel Prize work. So we are thinking if we can generate a similar letter for RNA, and then we can de novo sequence RNA by mass spectrometry. The base calling can be made just by comparing the mass differences between two adjacent letter fragments, and then comparing them to the known nucleotide or modification to read each nucleotide in a sequence. One way to generate a letter like this is to cut the RNA once, but randomly among the RNA. And this can be achieved by controlled formic acid hydrolysis. This will generate a two letter pool, 5' letter pool and 3' letter pool. Sequence can be read from each pool and they can reaffirm each other for sequencing. And this is our data looks like. If we plot mass against retention time, we can see two clear curve. Because the small fragments come out early with small retention time, big fragments come out later with big retention time. So you can say this to a sigmoidal curve. We can also differentiate the 5' letter away from the 3' letter because 5' letter fragments has a phosphated 3' ends that the 3' letter fragments does not have. So with this, we can achieve de novo sequencing of RNA using mass spectrometry. We can read this bidirectionally. The major advantage of this method is that we can not just base calling ACGU for nucleobase, but essentially all the modifications. As you can see here, we have two methylation. We can pinpoint exactly where they are location. However, this method also has limitations, mainly because it can limit it to highly purified short RNA. It will be difficult to handle complicated cellular sample because they have many RNA is a mixture. So the question we were asking then, is it possible for us to de novo mass sequence of cellular tRNA? We know tRNA is a difficult one to get sequenced by high throughput sequencing technology because they have so many modifications. Also, it's difficult for mass to sequence a DNA because traditionally mass requires a highly purified single tRNA sequence, which is very difficult, as you may have heard from today's talk. It's very difficult to get. So we developed some strategy over the years. For the interest of time, I'm going to mainly focus on one strategy. That's a mass sum strategy. Mass sum is a computational data separation strategy. Remember, we rely on formic acid to generate a ladder, a Sanger type of ladder. If we look at the any pair of this ladder generated by this one-color events, their mass sum is a constant, is equals to the mass of the RNA plus a water molecule. If you look at the 5' fragments 2 and their counterparts 3' letter fragments 7, their mass sum is a constant. Similarly, if you look at any pair of fragments generated by this one-color events, their mass sum is a constant. Because each RNA has the unique mass, so we can computationally separate all the letter fragments out of the complicated mass data for sequencing each tRNA in the mixture. So here is one of the example. So this is the tRNA, is the tRNA we purchased from Sigma address. As you can see, this is a mixture. The major components actually is not the full-length product. It's a 75 with again missing at 3' ends. Despite the physical difficulty to separate them, we can computationally separate all the letter fragments using the unique mass of this isoform, and then sequence each individual one. So with this approach, we can reduce the need of physical purification of a specific tRNA and allow mass sequencing any RNA in the mixture directly. And this all, of course, can help to increase the throughput of mass sequencing, allow mass potential sequencing, and even the number of RNA allowed by the instruments. And this also proved ways for large-scale mass sequencing of RNA. Another challenge for mass sequencing is like pseudouridine and uridine has identical mass. Mass alone would be difficult to separate them. So in order to differentiate pseudouridine and uridine, we treat RNA with CMC. CMC is going to selectively react with the pseudouridine. The mass is going to increase to 252. After the acid hydrolysis, their letter fragments is going to shift to the big mass region. And also because of the CMC complex is more hydrophobic. So after the acid hydrolysis, and the letter fragments is going to shift to the big retention time region. So you can see this is a curve it's branching here, but also the CMC conversion is not 100%. So we're going to see two curves. One is the original one, unconverted converted one. One is a new one that converted. All starting from this pseudouridine. So our data actually match very well. You can see we can see the branches as we expected, and this indicates where the pseudouridine located. And if there's more than one pseudouridine, we can see multiple branches. This branch feature actually is very significant because a branch in this 2D mass and retention time curve suggesting partial single nucleotide substitution. So a branch that we have seen here indicates the coexisting of two nucleotides signifying a partial RNA modification, which is difficult to study, as you heard today from another talks. So this is the one of the branches we have seen when we sequencing tRNA glue from Professor Chen Qi, his lab. He's going to talk more in his later talk. But you can see we can base calling the nucleotides. We find that A at position 18. We also find a methylated A, same size. And their differences is like 14,000 corresponding methyl A. So with this, we would like we can know the identity of the modification, the location of the modification. We can order further determine the stoichiometry of this partial modification. The reason for this is the mass signal we use for sequencing also carries relative abundance information. So we can extract ion chromatography and integrate them to calculate their stoichiometry. In this case, this is about 40% was 60% 40 of methylated A. And the reason that we can do that, I have to see, this is not an absolute quantification. This is just a relative stoichiometry of methylated one versus non-methylated one, because they coexist at the same condition, at the same size, So they can work as internal fragments, references for each other. And we also further verified this is the accurate methods. So with all this development, now we can directly sequence a full-length tRNA de novo. Again, this is the sample from Dr. Chen Qi's lab. You can see we cannot just read the full nucleobase ACGU, but we can read de novo read all the modifications as well. Among these 10 modification sites, 3 of them are not modified 100%. We further determined their stoichiometry as shown here. To our knowledge, this is for the first time we can de novo sequence full-length tRNA with single nucleobase and stoichiometric precision. And this, of course, can help to reveal the true sequence of RNA or RNA. And their tRNA has isomeric methylations. If the methylation is at a 2' position, and they're going to block our acid hydrolysis chemistry. So we know there's going to be a gap. So this is corresponding to like 2O methyl. Of course, there's a lot of methylation as well. And to differentiate them, typically is very challenging. Here, we just want to talk the efforts. We work together with Dr. Chen Qi lab. We take advantage of specificity of RKB enzyme. They are only going to selectively demethylate this 3 methylated nucleotides. So before RKB treatment, we can see this one curve. This is corresponding to methylated A at position 58. But after RKB treatment, you can see this curve almost completely disappeared, and we see a new ladder corresponding to regular A. So we know this is sensitive to this RKB enzyme. We know this is the M1A. Similarly, we also find in the position line, and this should be the M1G, not a non-methylated G. So with this capacity, we actually can help to address a bigger challenge right now in the field, potentially allow us to monitor the dynamics of RNA modification or even the dynamics of RNA. And this could help us to map modifications that the author has changed the stoichiometry in different cellular or disease conditions. And this method can also handle complicated samples like a total tRNA. So this is the data in the RNA we observed from each total tRNA. You can see each dot here represents one tRNA because the tRNA has a mass about 24,000 Dalton. So each mass here indicates a tRNA. Some of them can be matched with the one in the tRNA database, and those are shown in orange. But they also have the ones that they can't match with the ones in a database. Indicates they either have new modification or have new sequence. Of course, we want to sequence them to verify that. So we did acid hydrolysis, and this is our data, how our data looks like. This, of course, is very complicated. Our algorithm is still under development. We can't read the full length of each of those tRNA, but with our current method, we can read parts of those segments. So with this, we can do blast to find which tRNA that belongs to. For example, we find this belongs to tRNA ASP. With the information, we can find further more of those letter fragments. And though the letter in green are the nucleotides verified by our letter fragments, the letter in box are the modifications that are verified by our fragments. So one by one, we can sequence each tRNA. Of course, some of them are not modified 100%, and we further determine that stoichiometry. So with this approach, we can identify, locate, and determine stoichiometry of each tRNA in total in that total tRNA. And now we actually have a better algorithm. Since we published this, we can actually sequence even those the one that we couldn't find in the current database. So Of course, all this is just the beginning. There's so many things we need to optimize. So many questions we can ask. Natural question would be: is it possible to use mass spectrometry to sequence a lot of RNA beyond tRNA? With the capacity to sequence total tRNA with hundreds of different tRNA sequences, I don't see why we can't sequence a lot of longer RNA, like ribosomal RNA, or potentially messenger RNA. We are now testing this. We remain optimistic about the future using mass spectrometry to sequence RNA, and we are comfortable this can be handled on large scale RNA sequences as well. So we are thinking after optimization, we expect the sequencing, we in one single run, we can sequencing about 8,000 nucleobase in 30 minutes, I would say run. That means if we can run the instrument 24/7, our daily sequencing capacity can reach more than 4 million nucleobase using one single LC-MS instrument. So this is how the path we propose to sequence the first human RNA. With our capacity to sequence the tRNA to be with the true sequence of tRNA, we have a solid foundation for the tRNA known, and this will contribute to 10 to 20% of first human RNA known. If we can further extend this to larger RNA, like ribosomal RNA or even messenger RNA, we are moving close to sequence the first completed human RNA known. The sequencing capacity, of course, is not comparable to current high throughput sequencing technology, but as long as we can achieve the sequencing throughput like Sanger sequencing, that's significantly enough. After all, Sanger sequencing is the sequencing technology that being used for completing the first human genome project. Of course, a lot of methods like the high throughput technology here, they can help. They can provide sequence information of ACGU and verify size of nucleotides. But the mass spectrometry might be the core technology to complete the first human RNA project. So this is really exciting time for us working in mass spectrometry. I really, by working together, I think we can complete the first RNA, human RNA known. I really look forward to the day will come out earlier. So with this, I would like to thank the people who are working in this project and our collaborator providing the sample and also NIH for funding support. I will stop here. I'll be happy to answer any questions. Yeah.
Thank you, Long. That was a wonderful global framing of the problem and some brilliant progress on that frontier of the RNome. I think we have a time constraint. I have a series of questions I could ask, but of course that would be taking up time that would be taking away from our next speaker. So there are questions emerging in the chat. If you, Long, can aim to address those, we can stay on time and move forward with Dr. Garcia's talk.
Sure. Thank you.
Thank you very much. Okay, everyone. Our next speaker, keeping on time, is Dr. Ben Garcia from the Washington University in St. Louis, and he is going to present a story titled "Quantitative Mass Spectrometry Approaches for Characterization of RNA Modifications." Ben, if you're there, the stage is yours.
Okay,
There
thank
you are.
Let me just figure out how to share. Okay, getting there. Can people see this? Look okay?
Yes, looks great. Looks great.
Okay, let me just get my pointer.
You are in presenter mode now, so you might want to switch it over.
Oh, switch it. Okay.
Yep.
Okay, how's that look?
That's great. Perfect.
Okay, great. Well, thank you, Scott. Thank you to all the organizers for giving me an opportunity to just kind of give an update on some of the things that we've been working on in terms of developing mass spectrometry-based approaches for characterization of RNA modifications. I know you've already heard a lot of great talks and seen a lot of really cool, exciting new approaches. So hopefully some of the things that I'll show you might add on top of all the great work that's already been discussed. I do want to spend part of this time I have to discuss some things to do with global mapping, mononucleoside, and then work our way up to some of the efforts that we've been doing for sequencing RNA oligonucleotides as well. Okay, and so I don't think I have to give a lot of background on this, but my lab is mostly known for protein modification work, looking at protein post-translational modifications. And we've been interested in histones and other highly modified proteins because, there's a whole host of modifications that are involved in regulating gene expression and other processes. And there's enzymes that put the modifications, take them off, and proteins that bind to these modifications or are excluded when modifications are present. So our interest in RNA has kind of grown from the protein side because we see a lot of parallels from kind of like the histone code and the epigenetic world to RNA modifications, epitranscriptomics as well. You have several different types of modifications, hundreds that have been discovered on many different types of organisms and a whole host of enzymes that put them on, remove them, very dynamic, and many proteins that bind these modifications, important in a lot of areas. And we've seen a lot of great technology advancements in genomics to identify a lot of these modifications and quantify them. But as many in this symposium feel, mass spectrometry definitely can play a big role complementary to genomics for characterization of post-transcriptional modifications on RNA and looking at combinations of modifications on RNA transcripts as well. And so I'll talk a little bit about some of our efforts to just improve the global analysis of RNA. We've already seen a lot of really great work. But, there's still some challenges. A lot of things aren't really kind of set in stone yet. Everybody's doing a lot of different things. And we've kind of jumped into this world not knowing the best ways to do things and found that, even at the mononucleoside level, there's a lot of challenges there. A lot of people are moving to HILIC and other really fancy chromatography systems, a lot of different ion pairing reagents that you probably don't want to spray in your mass spec for too long. And so we kind of thought about this. Can we make an approach that would be compatible with the chromatography we know for proteomics, just reverse phase HPLC, acid-based, and still get pretty good results and separation? Typically C18 doesn't always give you the best separation of mononucleosides. But kind of borrowing some tricks from the oligosaccharide field, we decided to kind of derivatize ribonucleosides with iodomethane that has deuterium on it, so CD3I. And this is kind of the procedure to do that. We use a liquid extraction to get what we call permethylated ribonucleosides. And this has been a strategy that has been used heavily in the glycan field. It makes molecules more hydrophobic and allows better separations. All the hydrogen atoms on hydroxyl groups and amines, carboxylic acid groups will be replaced. And so when we kind of do this procedure and we've optimized it quite a bit, little tricky to get going, but not too hard. You can see here with native ribonucleosides, at least in our hands, we get crowded chromatograms, but with permethylation, you can spread them out quite a bit. And the interesting thing about this is that, of course, when you have different modifications, the position on modification matters and can be differentiated by the fragmentation pattern of looking at the endogenous methylation versus permethylated, the CD3 sites. And so here's just a series of modifications on adenosine. And you can see when you derivatize just adenosine, unmodified mononucleoside, with this reagent, and these are the, what you would expect. If you have M6A, you can see that, you derivatize the hydroxyl groups, you would derivatize one of the hydrogen groups. Here, and then so on and so forth. And so what ends up happening is that modifications that are isomeric or isobaric at the MS1 level are then now differentiated at the MS2 level because they will have different incorporation either at the base or the ribose of the deuterated reagent. And this has been really a nice approach that has allowed us to separate, some things that are kind of difficult to separate, different modified isomers, as well as uridine and pseudouridine, because as you can see here, they will have different incorporation sites of the permethylated CD3. And when you fragment, they will have different levels, different amounts of that reagent as well. We've also been working hard to put in a lot of efforts to create data-independent acquisition for RNA mononucleosides. I think everybody knows about targeted approaches where, whether you see the molecule or not, you can still trigger MS2 and quantify at the MS2 level. So a lot of MRM type of work. If something is unknown or low abundant, that might be harder to see, or you wouldn't even know what to look for. So, targeted work only works for things that you can expect or know. DDA, data-dependent approaches, kind of work as, an MS-MS. You get your full MS, and then that triggers MS-MS. So if something's known will trigger MS-MS. Something low abundant, or, sometimes you get it, sometimes you don't. So this is an approach that maybe for low-level PTMs doesn't always work. And so DIA is kind of in the middle. And so whether something is unknown or known or unknown, you are taking, masked ranges and fragmenting everything together. So you will get MS1, you let everything through at the MS2 and fragment it in a smaller mass range and stepping through. And so, definitely something that's known, you're going to detect in the MS2, and something unknown, you're going to see it as well. And so we've been kind of using this and just in general find that MS2 quantification is much more accurate than MS1. That's something that's been observed a lot in the proteomics world as well. So nothing really, super new there. But at the same time, this does improve the quantification of mononucleosides, and also allows for differentiation by looking at inspecting the MS2 and reconstructing the MS2 spectra. Differentiation of isomers quite easily, where maybe you wouldn't trigger a low-level species with a DDA approach. The other thing too is DIA data you could always go back and remine the data. And so maybe at this point in time, you take your data, you're happy with it, but later on a new modification on RNA is found, or you're talking to some people and now you have a hunch that something else might be there. You could always go back and remine the data. And so we've kind of done that and found a lot of potential either new modifications or things we just didn't expect. Sometimes they've ended up being artifacts of the processing, but other times we're not exactly sure. But this would work by taking your full MS, lining up the peaks that come from the DIA or the MS-MS, and then you can tell which ones are corresponding to which bases based on the peaks you see. And in this case, it indicates that there's a mass of 162, which would be, probably some type of single sugar unit, like a glucose. And so we've been kind of doing this, and this got us down a path, which I'll talk about a little bit more later. But when you go back and you kind of line up, the peaks and start looking at what MS-MS fragments line up within a base peak, you can see that there's several species that end up coming out here and allow you to identify certain modifications. And we started noticing that we started seeing signature ions of sugar molecules lining up with a lot of the RNA mononucleoside full MS data from our DDA data. And this kind of started getting us on a path of looking for sugars or oligosaccharides on RNA. And I'm going to get back to this at the end, but I'm going to switch gears now a little bit and talk about the RNA sequencing that we've been kind of doing in the lab. And so, we've been, interested in also developing, trying to see how hard it is to sequence RNA by MS-MS, and it's definitely a challenge, but it can be done. A really beautiful talk right before me on pushing the boundaries of this type of work. But when we first kind of started, we definitely found that, we could take RNA and digest it and run it, and we'd see a lot of species. But at the time, there wasn't a lot of great software available. And so the first thing we kind of did was kind of take the guts of the nucleic acid, take the guts of the OpenMS program and create a nucleic acid search engine. We just call it NACE. This was a nice collaboration with Hendrik Weisser and Oliver Kolbachter over in Europe. And here, we started kind of thinking a little bit more about, proteins have different corresponding isotopes than RNA, different fragmentation patterns as well, and started kind of, putting these into the search engine kind of feature to be able to do, some RNA shotgun types of omics analysis, mass spectrometry analysis. And with this software, it works pretty well. We're able to sequence RNA, identify modifications, and kind of, be able to map these to specific RNA sequences. At least in our hands, we found that our program allowed for identification of more RNA spectra, as well as allowed us to identify longer species as well. And so this was published a little while ago. It's kind of our first foray into RNA sequencing, but really sets up the approaches that we're using now. And so, and this program has continued to be updated by a former graduate student of mine, Sam Wayne, who is working with the MS Omics team. So I would definitely encourage people to kind of check out the program. Definitely works pretty well after a little bit of primer and understanding what you're doing with this. The other thing we've kind of been working on, kind of in the background as well, is looking a little bit at what can we do on the front end in terms of enhancing separations of RNA, because once you get to using C18 or other types of chromatography, the separations you can get, the quality is never as high as you can do with peptides. And so we've been looking at including this field asymmetric ion mobility mass spectrometry, FAMES, into kind of our workflows. FAMES works because it essentially separates out peptides, electric fields by charge. And so if you're thinking about charge, the longer something is, the more charge it's going to have. So you can see here as we step up the FAMES voltage, no FAMES, and then step it up this way, you can get different length oligonucleotides separated out. And it does kind of correlate well with like the different charge states. So you have different charge states of the RNA separated out. This not only boosts the MS1, but also the MS2. You can see here, no FAMES and FAMES. We get increased fragments with the FAMES device on. And this allows for, just better scoring, better detection of a lot of different modifications, quantification as well. And so I would definitely encourage people to use FAMES device if you have access to one, because I think this does help. And for low-level species, it helps get rid of some of the things that you might see in the background to be able to sequence those low-level RNA species pretty well. We've also been using porous graphite carbon as well for separation of RNA. PGC has been used heavily in the RNA world and for other hydrophilic molecules. But the way we're doing it is actually a little bit different. And so what researchers have found out a while ago is that if you put voltages onto PGC, you can create electrochemical modulated gradients. And in fact, this is how several RNA biosensors operate under certain polarity. You trap your RNA and then you can release them with the reversal of that polarity. And so we thought, well, what if we can incorporate this into kind of our analytical setup? If we have our normal C18 columns and a trapping column, also PGC, so this is all PGC, not C18. But if we put a voltage here on the trapping column, could we, trap and load RNAs and then release them with a voltage switch to have a second degree of separation? And so here's kind of a proof of principle experiment. We're holding the voltage constant here. And there's a few things leaking out because we are increasing the gradient. But then right here, we have the polarity switch and then another gradient, and you can see a lot more things coming out. Of course, this isn't all really great and separated, but it just shows that you can trap RNA in your kind of HPLC setup. And you can see here, once we kind of switch the voltage, you get the RNA released. And so we're able to kind of sequence RNA through this type of approach, just looking at different species that we're separating out with different voltages here. And overall, we feel that this electroelution chromatography is really nice, allows us to separate, by the different voltages, get more IDs. It is dependent on the RNA charge, of course. So then you're also separating out by the RNA length because you'll have more different charge numbers on different RNA lengths. It allows us to separate and characterize a lot of modifications, just kind of shown here with TNA as well. So it's definitely something that we're playing with, or we have played with, and we would like to keep optimizing. Right now, it's not fully a plug and play kind of system. You have to kind of do steps, but at the same time, this could be something that could be orthogonal separation for RNA. This has allowed us to identify a lot of different species that we've been, determining the different modifications, such as oligosaccharides, which I kind of mentioned before. And, this is something that has become of high interest because, of some recent publications that have found that small RNAs can be decorated with glycans, especially on the surface of cells. And so we've been working with Ryan Flynn, who now has his own lab, in trying to characterize, the N-glycans and O-glycans that are found on RNA. And so, workflows are pretty kind of similar to a lot of workflows. Sometimes we use, PGC, sometimes we use other things. You have to be really careful just to make sure that there's no protein contaminations. And so, this is something that, the sample preps are really, really important for this type of work. But, had a lot of experiences now, Ryan's lab as well, and it's given a lot of good pointers for this. And what we find is that the N-glycans that are separated and released from RNA are very different than, are different than the ones on proteins. There are definitely some overlaps, but there's, some specificity either way. And kind of in general, RNA samples are kind of enriched a little bit more with certain oligosaccharides, these fucosylated kind of RNA oligosaccharide molecules. And we've started mapping a lot of the oligosaccharide content on RNA, on these glycoRNAs as well, from various different cell types, but also a lot of different tissues across the mouse and even human samples and things like that. And so right now we're really interested in kind of continuing to further this. We've been able to identify some of the at least one of the linkages between RNA and oligosaccharides as well, the APCU. And this has kind of opened up a lot of new biology that hopefully people in the RNA world or on the glyco world will be interested in. So with that, I'd just like to wrap up to stay on time. We've been working a lot on enhancing and making the global analysis of RNA mononucleosides just more facile. This permethylation works pretty well, allows us to use a lot of the same buffers that we use, the exact same buffers that we use for proteomics. And so that has been very, very useful and also given us enhanced data along with the DIA work, which allows us to improve quantification and identify potential novel modifications on RNA. Been playing with the separations, both ion mobility as well as this new electroelution chromatography to enhance RNA modification mapping as well. And this is opening up new biology, especially on the side of glycoRNAs. So with that, just like to wrap up, thank the people involved. This is kind of one of the pictures of the current lab, funding sources, and I'd be happy to try to answer any questions if we have time. Thank you.
Wonderful. Thank you so much. Another tour de force example of your innovative approach to science. I really appreciate what you discussed. I have some questions, but I'll start with the first question that came online, which is how much RNA do you need for the DIA analysis?
No, it's not a lot. And I probably should know the exact numbers, but it's in a very low amounts. I would say probably yeah, low nanogram or lower. The there it depends which instrument you're doing the DIA on. So obviously you could probably even go even lower to probably pre-picogram level if you're running on one of the higher end kind of mass spectrometers, Astral or something like that. Most of the work that we're doing is either on kind of a Exploris 240 or an Ascend or even on a SciX Xenotoph. So maybe not the most sensitive instruments, but you get plenty of RNA from cells. So yeah, we usually end up with a lot more than what we need, and even allows us to do some separations of the RNA before we even inject in the mass spec. If we want to look at small RNAs, large RNAs, or some from specific compartments.
Another question coming from online. Does the presence of metal adducts help or hurt electroelution and FAME's separation of RNA?
Yeah, that's a really good question. I think for certain it will interfere with the electroelution. And in fact, when we operate this system, you do have to, kind of clean the system after a while because you will get kind of accumulation of some salts. And so, yeah, in terms of the FAMEs, that I don't know if we've really looked at that enough in detail to see if it would also impact the FAMEs, but my guess is it probably could as well. I think typically under the conditions we run, we don't see a lot of metal adducts, on our, all of this is nanoflow too, is what I didn't mention. So we don't see a lot of adducts. We try not to use a lot of salts, ion pairing reagents. But my guess is if the concentrations were high enough, it would affect both. Yeah.
Great. Great. And I think I'm going to interject one of my questions, which I thought might be helpful to the discussion, is this modification, this permethylation step where you can really increase the hydrophobicity of your target RNA. It's really clever and helpful. I just wanted to make sure that I understood how that method works. Is it like a neutral pH modification, or is it organic phase? How do you do that
No,
exactly?
it's I believe the reaction is a higher pH. I think at least probably around pH 8. And so we've only gotten it to adapt it to work for mononucleosides because at the oligonucleotide level, it probably induce cleavage.
Okay.
So it's a higher pH kind of,
But not too harsh.
not crazy harsh, no, but it is definitely higher. And I guess in terms of your second question, we use liquid-liquid phase separation because yeah, you can kind of separate the organic from the aqueous phases, and that's kind of allows us to kind of clean up the sample.
Great. And my second question, which is totally unrelated, is what is the current thinking in the field of the role of these glycosylations in RNA? What is going on there in your view?
Yeah, I guess it depends who you ask.
Yeah.
I think for a while it was, there was a lot of skepticism about this. And even when the initial papers came out, I think we were kind of skeptical too. But in working with Ryan, these things do exist. We definitely see them. We see the linkage site. We discovered that was a paper published last year in Cell. And so I think the role biologically is to mediate probably like protein-protein interactions and extracellular matrix kind of interactions. There have been some nice biological papers to show that they are important for mediating certain immune responses as well. But, I think this is such a new field.
Yep.
The next years are really going to, kind of, I think, explode with a lot new information on the role of these glycoRNAs.
Fantastic to hear your perspective. Thank you so much for your presentation and for your time. Wonderful
Thanks.
talk.